Okay, good morning, everyone, and welcome to our fourth Neural Engineering Symposium. My name is Dalton Dietrich, and I'm the co-director of the University of Miami Neuroengineering Institute and scientific director of the Miami Project to Cure Paralysis at the University of Miami Miller School of Medicine. COVID-19 has presented many challenges to our community and our scientists. I hope everyone is staying safe and well. In response to this pandemic, we've really upgraded our ability to communicate with our colleagues within our universities and across the nation and internationally. Through Zoom meetings and webinars such as this today, we're finding that we can still build alliances and initiate collaborations between the various colleagues. Today is our first virtual neuroengineering symposium, and we really appreciate everyone for being here today and participating. Thanks to the participants, and over 200 individuals will be participating in today's meeting. We're really excited to tell you that representatives from many universities throughout the nation, including University of Florida, Florida State University, Florida International University, University of South Florida, Max Planck, Washington State University, and of course, University of Miami are participating. It's a great turnout, and I hope everyone has a fantastic time. Now, the purpose of this meeting and previous meetings has exchange of information and to build alliances that can attack very, very complicated problems. The Neuroengineering Institute covers a multitude of different types of problems, basic translational and clinical research, and many clinical problems such as stroke, neurotrauma, paralysis, aging, cognition, and sensory, including visual, auditory, as well as balance. So the Neuroengineering Institute's purpose is to bring people together, but also attack and utilize new technologies. And there's so many new technologies in the field that neuroscientists such as myself can utilize to propel their research to the next level. Neuromodulation, brain-computer interface work, for example, machine learning, virtual reality, new biomarkers that we can put our cells in as we actually transplant into people. So it's a very exciting time in the neuroscience and engineering community. At the University of Miami, we felt that having an institute would bring our engineers and our neuroscientists together, and it certainly is working. Another initiative of the Institute and the symposium is to help recruit new scientists and new students into this very exciting field, to train the next generation of investigators to utilize these technologies to propel their research initiatives. It's really exciting to know that students coming into the Department of Biomedical Engineering, for example, are very interested in our master's program in neuroengineering. So things are really taking off. And again, we want to propel this research throughout the state of Florida and elsewhere. Meetings like this could never take place without a lot of help from a lot of people. We especially like to thank the University of Miami Clinical, Translational, and Science Institute for supporting our translational presentations today. I'd also like to thank Erica Suazo, who has helped organize this and previous neuroengineering symposium, and Ignacio, an outstanding AV specialist who's helping to drive this virtual event. Good luck, Iggy, today. Okay, now I'd like to introduce my colleague and friend, Dr. Sedigh Rajguru, who's an associate professor in the Department of Biomedical Engineering and a very important collaborator within the Miami Project to Cure Paralysis. So enjoy the meeting. Sedigh and I would really like to get any comments back from you regarding how we can actually improve the meeting next year. And I hope you have a fantastic time. Again, thank you for participating.
All right. Thank you, Dalton, and good morning, everybody. Welcome to the fourth annual Neural Engineering Research Symposium at the University of Miami. We're really excited to have you here. This year, we have a really great program that has 4 thematic sessions that will start off with computational neuroscience and deep learning, followed by emerging tools and techniques in neuroscience. And tomorrow we'll have 2 additional sessions on brain-machine interfaces and the aging brain. After which tomorrow we'll also have a poster session. We have received 30 entries for posters from around state of Florida, all the different universities. Really exciting presentations that have been posted on Box, and a link has been shared with all of you, so you can all view those. They'll be available for 30 days for you to view, and we'll also have awards for the posters. We'll have 5 top awards, 3 to be given out for $250 each by the University of Miami's Institute for Neural Engineering, and 2 for $250 in translational neuroscience to be given by the Miami CTSI. So I really want to encourage you to engage with us through this virtual platform, through the Box platform that we have. As Dalton said, we always welcome your feedback on how we can improve this, how we can change this, what kind of new things we can bring to the neuroscience and neural engineering communities. This year, I really want to thank our student and trainee organizers. So Shu Pan, who is a graduate student in computer science, Hannah Radabo, sorry, I shouldn't have pronounced that properly, but graduate student in neuroscience, working with Dalton actually, and Raquel Sangaletti, who is an assistant scientist in otolaryngology. They are the ones who have actually led the entire program, decided the themes of the sessions that we'll have today and tomorrow, and really contacted all the speakers and organized this. So I really want to thank the 3 of them for leading this entire symposium and organizing it. With that, I'm going to turn it over to Shu to kick off the first session on computational neuroscience and deep learning.
Thank you, Dr. Rajguru. Welcome to the 4th Annual Neural Engineering Research Symposium, Session 1, Computational Neuroscience and Deep Learning. My name is Xu Pan. I'm the moderator of this session. So in this session, we will hear 6 exciting talks. Each talk is 10 minutes. Q&A and a 30-minute panel discussion will be after 6 talks. You can put your question and discussion topics you want to hear in the Q&A. So please use the Q&A function. For speakers, it would be great if you can time yourself. I also prepared a sign of 1 minute left. Hopefully this also helps. So without further ado, let's welcome our first speaker, Dr. Madineh Sedigh-Sarvestani. She's a postdoc fellow in Dr. David Fitzpatrick's lab at Max Planck, Florida Institute for Neuroscience. You can share your screen now, Madineh.
Okay, great. Great, good morning. So thank you for the opportunity to speak, and thanks to Shu and all the rest of the organizers for putting together what looks like a really great symposium. I'm going to tell you about retinotopic maps today. These kind of maps are really a transformation of the visual field, or equivalently the retinal surface on the cortical surface. We've mostly studied these maps in primary visual areas because they're larger and more accessible, and the transform, or the maps, are pretty simple in these areas. So you take the 2-dimensional retinal surface and you basically transform it in a linear fashion to the 2-dimensional cortical surface, allowing for some deformations for curvature and the shape of the cortex. Beyond V1, we don't really know much about the details of these retinotopy maps, in part because these higher order visual areas tend to be stuck in folds in the primate brain, so they're hard to access. This is why I'm going to tell you about these maps in higher order visual areas like area V2, the secondary visual area that wraps around V1, and this little critter, the tree shrew, that I've been studying. It kind of looks like a squirrel, but it really should be thought of as more primate-like because it has a very well-developed visual system with primate-like features. And the important factor here is that it has a smooth brain. So all of these higher order visual areas are accessible to me for imaging. I'm going to measure retinotopy maps in awake animals using calcium imaging. So I'm going to show simple stimuli to these animals, which are bars that are moving in visual space 1 dimension at a time, either in azimuth, left or right, or elevation, up or down. And I'm going to measure the activity while the animals are viewing these stimuli, and then use a simple color code for either the x-axis dimension, azimuth, or the y-axis dimension, elevation, to produce the maps on the cortical surface. So in V1, you can see they look very simple. They basically follow the color map because of the linear transform. And unfortunately, I can't measure all of V1 and V2. So instead, I'm going to put a small window over the border of these two regions and look at a little bit of each of these areas simultaneously. And from here on, I'm going to rotate this window 45 degrees clockwise. So the azimuth dimension runs left and right as it does in the visual field. So here's some data. Let's sort of step through it. These are the top row here of these black maps. V1 is on the right, V2 is on the left, and the top bar is responses for a bar in visual space that's moving up, so moving up in elevation. And you can see the bump of activity in the cortex basically follows a mirror version of the bar. And that produces a pretty simple mapping that's basically just a reflection of the color map in V1 and V2. For the azimuth direction, things look pretty simple in V1. You have a bump of activity that's basically following the bar. In V2, that looks very different. So whereas in V1, the color map basically looks like a small portion of the color code here because the rest of it is outside of our window. In V2, there's a couple of things going on. First of all, you have multiple regions that light up. Secondly, they're rotated 90 degrees relative to the activity patterns in V1. So the map in V2 just doesn't look like V1 altogether. So I spent really almost 6 months trying to figure out the transform between the visual field and V2 that would produce this pattern. In V1, the transform is simple, it's linear. You can tell just by looking. But it is more complicated in V2. And it turns out that if you have this sort of sinusoidal snaky pattern of the visual field, and you use that to transform to V2, you get maps that look quite similar to the data. So I'm going to show you this animation where I'm going to step through the visual field along this path, and each time put a stripe across V2, this thin region, and build the map up. So I'm moving here along the x-axis in azimuth. Then I turn around in elevation, azimuth again, moving in elevation. And I'm building these stripes that look similar to the azimuth map because my color code here for the visual field is the azimuth color code. If I use the same transform, but now I just change the color code to the y-axis, the elevation, the transform again produces an elevation map that looks similar to the real data in V2. So it really looks like there's this complicated sinusoidal transform that's defining the relationship between how the visual field is encoded on the cortical surface. And of course, this is a hypothesis, but you know where I'm going with this. I'm going to show you that the data supports this. So we can see the signatures of the transform by projecting the elevation and azimuth, or projecting the curve in the visual field corresponding to a line on the cortex. So in V1, if I put a line on the cortex, sort of perpendicular to the change in elevation, because the transformation is linear, that line is going to produce a line in the visual field that's changing across one dimension, the y-axis at a time. In V2, a parallel line is going to produce the snaky pattern because the transform is very different. It's sinusoidal, and it's changing in both dimensions at the same time. And these oscillations between regions towards the left of the visual field and regions to the right correspond to these stripes. So you're doing the zigzaggy pattern of the visual field, and that's how you build up the map, whereas normally you would just do a one-to-one grid transformation of the visual field in simpler visual areas. So the obvious question here is, why does the brain choose to use this much more complicated transform in V2, a one-dimensional transform, where both maps are encoded along the same sort of long axis of this thin region, whereas in all other maps, basically, we've used a two-dimensional transform with orthogonal axes. And I've sort of given away the answer. It has to do with the elongation of area V2. So area V2 is relatively anisotropic. It's thin and long compared to primary visual areas, which are isotropic. And this, so here I've produced sort of a two-dimensional version of a would-be V2. And you can see that it's squished along one dimension, along the short dimension. So if this is the dimension that encodes azimuth, your visual perception resolution for that dimension of the visual field would always be lower than it would be for elevation. So let's say you tilt your head, now you're going to see a different view of the visual world because you don't have round receptive fields in visual space, sort of. To see this, I've simulated a two-dimensionally encoded V2, and here you're looking at little pixels and their corresponding, the spread of their points in the visual field. And the fact that they're all sort of along the same dimension shows this lack of resolution in one dimension of visual space. What you get with this one-dimensional transform is that now you can have resolution. So again, I'm plotting pixels in the map and showing the point spread function or the region of visual field they correspond to. Now you have all shapes of these spread functions. So you have resolutions in different portions that are better in elevation or azimuth, on average round, and you're not locked into this low resolution space. Okay, great, I have a little bit of time. And then once we've switched to the 1D transformation, there are many ways you can use a one-dimensional transform to cover a two-dimensional visual field. Why this particular sinusoidal pattern? I actually thought this was a fun part of the data. So if you take the gradients of each of these axes separately and you plot them against each other, you can almost see this in the path itself in the transform. They're out of phase. And this happens because you're either changing azimuth primarily or elevation, but not both. So the rates of change are out of phase. And it turns out that this out of phaseness in this one-dimensional transform is the optimal way to cover a 2D visual field. So if they're changing in phase, you're not covering as much of the visual field. If one of them isn't sinusoidal, thank you, 1 minute, you're having lower visual field coverage. And it turns out that this is a known concept in robotics literature. There's path planning studies that show for a one-dimensional operator, like a vacuum robot or a lawnmower, you want to take this sinusoidal pattern. So the brain is sort of doing the optimal encoding in one dimension. So to summarize all of this, I've shown using the tree shrew that optimal visual field coverage and resolution objectives can give rise to really drastically different retinotopy maps or transformation of the visual field on the cortical surface. And this is important because for decades we have relied on the assumption that cortical maps always look simple and are always a linear transformation of the retinal surface. And the relevance for this for modeling and building up sort of simulations of the brain is that now we can sort of understand that the objectives are important, but not the actual orthogonal characteristics. So in our models of the brain, we don't have to have this enforced constraint that the retinotopy models have to look like the retina. And the other way this is relevant for our understanding of how we build visual areas is that now we've shown that with simple brain wiring between visual areas, you can produce a complicated sinusoidal transform from a simple linear transform. So retinal wiring, or I'm sorry, wiring between cortical areas within a visual hierarchy can be very complicated and achieve complicated transforms. And thanks to all the people that contributed to this work, especially my PI, David Fitzpatrick.
Thank you, Madineh. Our next speaker is Joshua Bowen. He's a graduate student at the University of Miami Computer Science Department. He's my lab mate. We work in Dr. Odelia Schwartz's lab. You can share your screen now, Josh.
Great. So I work with Dr. Odelia Schwartz in computational modeling of the brain, particularly the visual cortex. I'm a PhD student here. So I would like to start off with just a basic question about what we're interested in doing. We're interested in understanding what the brain is doing, but there's a lot of ways we go about doing this. So I'm interested in the computations we might be able to abstract from the brain that give rise to the rich visual experience that we see in images. So when you or I look at this image, we see people walking around, trees, all sorts of objects. So you don't get this from mere light intensity. If you think of like the pixel values from a camera, you don't get this understanding of people walking around, trees, and all these kind of rich objects. So how is the brain able to perceive all this information from simple light intensity? Maybe there's a neural representation that allows for memory. How is it able to do this? And sensory processing in general. So what some people like to say is you see with the brain, you don't see with the eye. So what is going on that allows for all these mechanisms? So there's a few ways we go about trying to understand these computations. So there's two popular approaches. This by no means an exhaustive list, but there's the stimulus-oriented approaches and goal-oriented approaches. If you speak machine learning, this is kind of analogous to unsupervised and supervised learning. So stimulus-oriented approaches, this is trying to derive an image model solely based off of the stimulus information. And this includes things like natural scene statistics and efficient coding. So either trying to exploit the statistics of natural images or trying to build an efficient model of what you're representing. This is presumably because the brain is in the environment and it has limited neural resources. So it's probably exploiting things like this. The goal-oriented approaches, this includes things like deep learning that try to derive a model not just from the stimulus information, but from what are called labels that can be associated with the stimulus. So the image includes an image of like a dog, the label might be dog, and so on. So these models have a hierarchical layered approach, and you can see they build up higher level representations from lower level representations. These are particularly interesting because they're loosely based off of what we see going on in the brain. But by no means like the perfect model, but we see something like V1 going to V2 in this block diagram on the left, and we see V2 going to V3, and there's all sorts of connections. So maybe these layers and these models can correspond to things like V1, V2, and V3, and we could have this kind of compartmentalization of tasks, so we kind of just understand one layer at a time, or one region of the brain at a time. So where should we start with these pretty complex models? So we could start in V1, the primary visual cortex, but this is pretty well understood. We have this idea of sparse coding, that its goal is to model the sparse activation of the brain, so only a few number of neurons being active at any time. So it tries to do this by coding for images as a sum of images or model representations from a huge set. So this includes, if you train the model to find the best set of images, you see something like you see on the right, these Gabor filters. And if you remember from Hubel's and Wiesel's experiments, they found these bars of light that were well modeled by Gabor filters. So this was pretty exciting back in the day, 1996, and it's generally considered a decent model of V1. But there's a lot less understanding when it comes to V2, the secondary visual cortex. So maybe we can start here. So, and I also want to talk about how can we judge whether a model is performing well or performing poorly. So people typically check how the model's responses can correspond to neural firing rates. But we could also check how well these models perform at visual tasks. So like I was saying, we have like perception, memory, and all these kind of things. Well, we also have things like inference. So how is the brain able to infer information that it doesn't see? So if I hold my hand up in front of my face and I occlude something I can't see, I still have a decent idea of what's there. I just, unless like maybe I'd never seen it in my life before, but it's not compromising to the entire image. So how is the brain able to infer these missing information? You also have things like content generation, so imagination. So if I told you to like imagine a dog sitting at a laptop with a coffee mug and glasses on, you could probably do that even though you probably don't see that very often. But so what is going on? How are all these representations combined to give you something like this? And of course, there's image classification, which is a common one too. So my work is going to focus on the image inference, and we're also going to do image classification. We're focusing on inference because we believe there might be a mechanism for inference with sparse coding. So if we think of the sparse coding model, as we increase sparseness in these reconstructions of images, we get higher error in trying to code for images. So these model representations that sum together, they're not going to do as good of a job. This means that the individual model representations need to do more, and the error allows for more room for information to be introduced. So this could translate to more inference. And as you do higher level sparse coding, rather than just in V1, but in V2, you could do inference over a larger receptive field or larger space, the image, so higher level stuff. Here's our V2 model. So we modified the approach of Hosoya and Heber-Riemann. It's pretty standard for V1 and V1 complex. So we see that they start off with Gabor filters, they convolve with an image, and they get V1 simple cell responses. They do a computation that makes them phase invariant to model the phase invariance of the V1 complex cells. And then they do a PCA for the dimensionality reduction technique. And they did this to constrain the receptive field sizes of the V2, the subsequent V2 model units. And then we follow with sparse coding. So in their model, they did something called independent component analysis, which is very similar, but our inspiration for sparse coding is from this inference mechanism. So we have two experiments here. So one's a classification experiment, the other is the inference experiment. So the classification experiment, we try to do simple things. Here's two data sets that we use. There's 3 in total. So we try to classify the angles between line segments, like you see on the left, or we try to classify texture classes of like brick walls, rocks, sand, all this kind of stuff, and see how well the model can do that. We train a linear classifier on top of V2 responses. For the inference experiment, what we do is we remove information midway through the model, and then we see how well can this information be inferred. So for the classification results, we see that the independent component analysis model that was done by Hosoya and Heber-Riemann, that performs the best, but with sparse coding not too far behind with its best configuration of sparsity. If we increase the sparsity to get this inference kind of mechanism, we see the error increases significantly, but there's not a lot of inference that needs to be done here, and there's little reason to do that. So maybe we could just consider the best value of sparsity for this task. But we want to consider what happens now when we go on to an inference mechanism. So you see the image here is an image of an elephant. We remove this spot in the middle with that you see kind of like an elephant's nose on the mod column right here. This is showing you the different stages of the visual processing pipeline, the V1, V2, all that stuff we should see before. So if we remove a region in the center and we get that black spot there, we want to continue through the model and then to see if something like ICA sparse coding can kind of infer what was missing there. So ICA kind of preserves that information, but with sparse coding, we see that we have some kind of completion done there. We see that the elephant's nose was mostly restored with a higher value of sparsity rather than a lower value of sparsity. We see a better reconstruction. And if we take a look at a few images, we could see a similar story with more sparsity helping. And if you look at the third row, you see this is like a low contrast, a low spatial frequency image. So this is kind of like where we got the idea. Could there be like a basis function or a model representation that just codes these kind of low contrast regions? Another idea was, or another interesting thing was the car reconstruction on the bottom, where we see 2 different reconstructions of cars. So that was encouraging in terms of the model. So in the future, we might try to do occluded image classification to try to exploit this inference mechanism. We might have an application image compression because of the nature of removing information from the model. With nonlinear generative models, like nonlinear sparse coding models, we could have a more powerful sparse coding model, maybe something closer to V2 or other regions. And we also want to check correspondence with neural data like fMRI or something. So I'd like to thank my advisor, Dr. Delia Schwartz. She's a professor here in the computer science department. Collaborator of ours, Dr. Luis Sanchez-Giraldo. He was a postdoc in our lab, and now he's a professor at the University of Kentucky. And also suggestions from Dr. Bruno Salson at UC Berkeley.
Thank you, Josh. Our next speaker is Dr. Josh Principe. He's a distinguished professor and runs the Computational Neural Engineering Lab at the University of Florida. You can try to share your screen now, Dr. Principe.
Good morning, everyone. This is going to be a little change of pace. We are going to talk about deep learning and information theory. And I thank Odelia for the invitation. I hope you enjoy my talk. It's going to be slightly different, but I'm going to focus on modeling. And so let's start. There are basically two strategies to optimize performance of signals in noise. The two approaches, one was proposed by Shannon, and basically what he tried to do, he tried to use encoding and decoding strategies to exactly be able to reconstruct the signal by getting rid of the noise. While Wiener perspective is the one that we normally use in deep learning, is very different because we are giving only the signal, so we don't know anything about the statistics of the data, but we are given the signal, and we would like to find optimal projections such that we are going to minimize the noise during this process. And so these are the two fundamental approaches. We normally in deep learning, as I said, use the cybernetic approach that is basically given by Wiener. But here I'm going to also tell you how we can blend in the ideas of Shannon in deep learning. But first, I'm just going to give you a very brief definition of what is entropy, because entropy is really minus information. And Shannon was the first one that defined uncertainty on random variables. And he did it by simply taking the expected value of the log of the PDF of the data in discrete time, in discrete amplitudes. If it's continuous variables, it's just an integral. So this quantity is very similar in concept to the Boltzmann entropy, but Boltzmann entropy is associated with physical systems, while here we are talking about random variables. But its most wonderful definition is the definition of mutual information. Mutual information was defined by Shannon also, and it is really a measure of the statistical dependency between two random variables, x and y. And so if I'm writing it for continuous random variables, and you see that you also have the expected value, because this double integral of the PDF is an expected value of the log of the ratio of the joint distribution and the product of the marginals. And if you look at this, this ratio here, this is very similar to correlation coefficients that we all use and abuse in statistics. But here notice that we are talking, working with the full statistical information about the data, because we are using the joint distribution and the marginals. And we can rewrite this equation. We can prove that this is also can be written as a function of entropies, as the entropy of the marginal x, entropy of the marginal y, minus the entropy of the joint entropy of x and y. So this is going to be important because they are concepts that are associated with information, as you can see, because remember, entropy is minus information. So when you increase entropy, you decrease information. When you decrease entropy, you increase information. If we go now very briefly on optimization in neural networks, this is a very old theory. Of course, we only know what happens in the last 25 years, but this started long ago by optimizing functions. And the earliest work was by Pontiagrin, that told us how to optimize deterministic functions in functional space. Then much more interesting results for us is when let's assume that our data is stochastic. And so in the stochastic case, if you want to find an optimal of a function, what you do, you take the expected value of the f of x. Okay, and this gives us the Hamilton, Jacobian, Bellman's equations in control theory that are very important, but is basically the thing. But Zipkin, that is one of my favorite cyberneticists, or machine learning guys, if we call it with the conventional nomenclature now, said that the interesting problem is none of the above, but there's one when we are trying to do this optimization adaptively. That means we don't know neither f of x nor the statistics of the data. So we are in the process of, we have to estimate statistics and then find the optimal functions that we also don't know. So this is really the fundamental, the most difficult problem, and the one that we humans and animals in general, biological organisms, solve in the real world. And we are still learning how to do this optimally. But this is important for us to understand these three types of problems. And in fact, what my work has been for the last 15 years is exactly to bring information theory to help us learn in the adaptive regime. So this is the block diagram of supervised learning. Everybody normally sees this. You see, you receive from the world data pairs, X and the labels of targets. You have here an adaptive model that is a functional is a functional function of some parameters that you can train. You create an error by subtracting the targets with the output of the system, and then you normally put here mean square error. But if you do that, so you are just simply using, you know, second order statistics of the error signal. So you are wasting a lot of potential information. And so we should put here an information measure. And so this is exactly, we should use, for instance, the minimization of the error entropy if we really want to work with the full definition of the error. And of course, then you have your learning algorithm that normally in machine learning is gradient descent, but this is basically the same thing. I'm emphasizing here is the cost that you should use. In terms of mappings in neural networks, and this is one of the difficulties in deep learning, is what are the networks doing? And it's we think it's a black box, and I'd like to change your idea about that, because really it's not a black box. We know exactly what goes on because we create the system, we create the nonlinearities. And so if you have a very deep network with, let's say, 100 layers made of the same elements, each element does exactly the same thing. And so the question, the interesting question is, what does an element, what does a unit in a neural network compute? This is really what I think is interesting. And the best way to look into this from the point of view of geometry is to look at neural network layers in pairs. You have the first weights of the first layer. These weights allow the system to come up with these signals that I call the phi1, phi2, phi k. So just think that these signals really are axes of a projection space. So basically what you are doing when you feed in the data and you adapt the weights, what you are doing, you are changing the location of this projection space in different ways. And what are the ways? Of course, are the ones that are going to minimize your error, right? And so basically these weights are going to rotate and translate the hyperplanes. You can think of this as an hyperplane. It can also be a manifold, but for us it's probably simpler to think of hyperplane. So basically these weights in the first layer are basically going to change the location of these hyperplanes. While the weights on the second layer, they just project the desired response on the hyperplane. And this figure here shows exactly that. So this is the space of your data in the blue, the space, the hyperplane that the system is rotating during learning is like this, and the purpose is exactly to decrease this distance between this hyperplane and your targets. So this is what neural networks do. So this is practically, conceptually easy to understand. The issue is how can we quantify the performance of these projections? This is really the fundamental problem. And if you're interested in this, in one of my books in 2000, you can see this explanation very well. And this is exactly where I think information theory can bring is quantitative understanding of this very geometric idea that I just told you. And for this, I have to define first what is a Markov model. So a Markov model is a stochastic model that characterizes randomly changing systems. With a very important assumption. It says that the information about the past of the signal is always represented by the current state. So you decouple basically the history of the signal from, and summarize it in the current state. So this is what is called Markov assumption that really simplifies tremendously the use of statistics in time series analysis, because otherwise you'll have to be always doing a conditioning on the past of the data. So this would be impossible to do in practice. And under this Markov assumption, if I have three variables, x, y, and z, you see that if this joint distribution obeys the Markov model assumption, I can write this as the product of p of x times p of y given x times of p of z given y. And if that is the case, notice that z is not dependent on x. And so you have this data processing inequality that is a very important thing that we normally forget in practice. That is the mutual information between x and y. So mutual information between x and y is always larger than the mutual information from x to z. And so if any system that you have, if you process the data, x and y, there is a processing step, you are already losing information about the original data. Of course, we do this very often because the data is noisy and we would like to decrease more the noise than the signal. But this is a very fundamental thing that we have to forget, is that when you process the data, you lose information on the data. And this is exactly given by this expression very nicely. And Tishby proposed this concept of mutual information to create a projection space inside of a neural network. So let's assume that this is a very long neural network and a very deep network. And what he says is that it is very nice to look at the mutual information from the input up to the input of a certain layer and map it with the other axis is the mutual information between the output of this layer to the output of the network. And so you have as one axis I of x and t, input up to the input of the layer, and then you map it with the mutual information of the layer to the output of the system. And it turns out that during learning, there is a trajectory that each one of the layers of this network, is going to take. And this gives us now the advantage of this mapping here is it is a scalar mapping that I can estimate every point, because I can estimate from the data directly the information, this mutual information and this mutual information. So this is really the fundamental idea that I would like to discuss for you, is that there are ways that we can now come up with quantitative information about the network, how the information flows inside the network, okay? And I can show you that there is a lot of interesting things, because there are effectively, when you train a network, there are two Markov chains. I don't have time to go through this. I just would like to show you that for this network with 7 layers, very large network, that was trained for the MNIST, we can now show exactly what is the decay of the mutual information across the layers of the network. And also, there is, as I said, another data processing inequality that comes from the errors. The errors in the network also also obey a data processing inequality, so decrease from the output to the input of the system. So as you can see, you can use these curves to see how well your network is learning, and you have to be really careful of not going to too deep networks that don't have any information about input. So this is something that in deep learning, sometimes people think more layers are always better, and it's not the case. We have to be very careful to train deep networks. And I think this is basically the message. So the conclusions is we are using information theory to understand the dynamics of learning. I had to give you a brief explanation of really the details of the theory, but I'm very confident, that this is going to be a very important way to help us design and choose parameters in deep networks that are so important in our research goals. Thank you very much for your attention.
Thank you, Dr. Principe. Our next speaker is Liang. She's a graduate student in Dr. Mingzhou Ding's lab at the University of Florida. You can go ahead and share your screen.
Okay, so can you guys see that?
Yes.
Yeah, so I'll start. Thank you for giving me this opportunity to represent our work in imaging trigeminal neuralgia pain and insights from combining the deep learning with grander causality. So the first way is, what is trigeminal neuralgia? It is a severe and disabling chronic facial pain condition which may last for years or even be lifelong. And the incidence rate in the United States by the current research is in the average of 100,000 people, there's an average of 4.3 people who got STM pain, and the incidence rate for female is larger than male. However, there's not a specific reason why this happens. There are several treatment options for that, for example, the microvascular decompression. However, the response to the treatment is highly variable. Some people may even got a surgery, but after years or after months, they still have this TM pain. In thus, this is an urgent need to better understand the mechanisms of TM pain, which may provide the more novelties for the treatment options. And so this comes to our two specific aims. Our aim one is to identify the essential regions in the brain that underlie the TM pain using the deep learning. And thus, this comes to our aim two, like how these regions are interacting with each other. We would like to characterize how these regions interact to produce the TM pain perception. So let's come to our experimental paradigm first. We will record the simultaneous EEG fMRI data simultaneously. And the first session is to taking the resting state for the patient for 7 minutes. And after that, they will have a 1 minute of pure resting. We do not have recordings. And after that, we will have the pain tracking sessions for continuous 20 minutes. In this 20 minutes, the patients would have a tracking ball and they can continuous monitoring, recording their real-time pain scores, which can be changed to a range of 0 to 100. And after that, we will record the T1 image for 5 minutes. And finally, we will record the DTI image for 17 minutes. You can see the below figure, there is sample pain level fluctuations for a specific patient. And we can see there's some kind of pain curves in the recording pain scores, in which we'll set the pain onset when the patient's pain comes to the highest curve, for example, in the 380 seconds. And after that, we'll show the methodology for our deep neural network. We modified the original RANSNet 3D the RANSNet network to the 3D volume by modifying the convolutional filters to 3D dimension. And also we adding the non-local block, which will add different weights in different locations. And we'll set the whole fMRI sequence as for each time step, we'll set them as an independent fMRI volume. And after all the data has been pre-processed by SPM, we'll feed the data into the RANSNet to our proposed network. And after generating the feature maps, we'll modify the final fully connected layers. We do the two strategies for predicting this pain. And first is the pain level prediction. We divide all the pain scores into 3 classes with low pain, high pain, and moderate pain. And another strategy we use is to predict the pain class prediction. The pain class prediction is one for continuous prediction, another is for the class prediction. And all the loss functions have been modified. After that, we use the GradCAM method to help us generate the heat maps and changing the heat maps to the anatomical space and do some thresholding. And finally, we'll average across other subjects. So for these two strategies, we'll generate 2 heat maps together. And for the robust analysis, we will intersect these 2 heat maps and to give us the final analysis of which regions are definitely activated. So this is the sample experimental results from our 2 strategies. The left figure shows the ROC curves for the pain level classification. We can get an average accuracy of 0.321. And the right figure shows the predicting pain scores versus the target pain scores. And we can have the R value of 0.838. That means the deep neural network can definitely help us generate some kind of the data distribution. And thus, we'll show the intersection heat map. This is the output of the intersection heat map. The 2 most regions we would like to see is the thalamus and DACC, which are the most essential regions in the original pain matrix. there are also some other regions showing up like insula, and we only point out these two regions as they are the most two important. So why we're focusing more on the thalamus and the ACC? We all know that this is the detailed introduction about the detailed location for the thalamus and the ACC. We all know that thalamus is the key structure linking the nociceptive sensory input to pain perception, and the ACC is the key structure for pain perception. So after we have set some kind of regions of interest, what kind of relationship between this information comes? In that way, we point out the granular causality. So what is granular causality? The granular causality is I will give a short introduction. It is a mathematical method to estimate the relationship between two time series. So for example, we have the one time series x1, x2 until xt, and another time series y1, y2 until yt. So how do we compare what kind of relationship between that? We will build two autoregressive models, in which one we call restricted model, which contains only the xt information inside us, and another we'll call the unrestricted models, which contains not only the x information, but also the y time sequence information into this. And we would like to compare the variance between the two residues for the two models. If the variance of the restricted model is smaller than the variance of the unrestricted if the variance of the unrestricted model is smaller than the variance of the restricted model, then we denote that the y time series has the granular causal influence on the x time sequence. So this is the mathematical methodology behind this. And this will show our GC results containing this. So in figure 1, you can see that we set the pain onset when the patient's pain comes to the highest curve, and we'll choose some specific time periods beforehand and afterhand, and set them separately as the pre-pain and pain period. And at the same time, we also choose another time period far away from the fluctuations, as setting them as the resting period. And thus, we're calculating the GC values from the thalamus to the ACC in these three periods. What we got is in the pre-pain and pain periods, the GC value from the thalamus to the ACC increases a lot and higher than the resting period. That means the information sending from the thalamus to the ACC increases a lot, even before the pain attacks happens, which is quite interesting. And otherwise, we also compute the GC value with the patient's daily experienced pain level, which is like shown in the patients would record their daily experienced pain level previously in a questionnaire. And we also build a simple linear regression between the two GC values. One is the DACC to thalamus, another is the thalamus to the ACC. And we found that only from the thalamus to the ACC, we have a p-value smaller than 0.01, which means the information sending from the thalamus to the ACC can help us to predict the patient's daily experienced pain levels, which also agrees with the previous result. So in this way, this is just a short summary. We have modified some DNA network and used that to help us predict the pain scores as well as the levels from the fMRI data. And after we have well-trained this deep neural network, we found that the thalamus and the ACC and insula are definitely shows as the signature centers of the TM pain, which agrees with the current research. And our second aim found that from the thalamus to the ACC, the GC value increases a lot, even before the TM pain attacks happens. And also from the GC value from the thalamus to the ACC can help us to predict the patient's daily experienced pain levels, which may provide more novelties for the future treatments. And that's all. Thank you.
Thank you very much, Yun. Our next speaker is Dr. Zheng Wang. He's an assistant professor at the University of Miami, Computer Science Department. You can share your screen now.
Today, my presentation's title is Studying the Regenerative and Non-Regenerative Neuron Cells from the perspective of 3D genome structures. So at first, we're going to mention two types of neuron cells. The first one is called regenerative. Second one is called non-regenerative. So basically, my collaborator, Dr. Vance Lemon and John Bixby at the University of Miami, they found out that some of the neuron cells can regenerate after being injured. Some of them cannot. So they are trying hard to find out why, genetically, what causes the regenerative and non-regenerative neuron cells. And for me, my perspective is to look at these two types of neuron cells from the perspective of 3D genome structures. These are not something we have already done, so I don't have any results to show you. But this is something we plan to do, and we are working on that. So you're going to see a lot of proposed research. I'm going to talk about 3D genome structures first. So this is a picture showing you the different levels of DNA. At first, we have the most detailed view is the showing the base pair, AGCT. And then you can have histones and DNA wrap up. And then if you keep zooming out, you're going to eventually see this kind of X shape. Everybody knows this X shape for one chromosome. But when the cell is not dividing, usually you don't see this kind of X shape, but some kind of like a ball structure. So our goal is trying to find out the 3D structure in this level. Not this one, but this one. These are some of the methods people have been trying to find it out. From 1970s all the way to 2009, there's a method called Hi-C was invented at that time, which can detect all versus all contact information. For example, if you have one point here, another point here, if these two locations, they are spatially close to each other or spatially proximate or in contact, then this experiment method can capture that. We call it one contact. Okay, then this diagram basically very briefly show you what Hi-C does. You have a chromosome, a piece of chromosome, a part of the chromosome. Then we cut it and we put some biotin, some chemicals, and then we can ligate. And after you ligate, you can see that this red part is connected with the blue part, although they are sequentially far away from each other. Because they are in the space, they are very close to each other. That's why it's possible that these two connect to each other. And then you have something like this. And then we cut it into segments. We got this kind of segment, this kind of segments. Blue and red, for example, this one, blue and red, sequentially they are far away from each other, but because they are close to each other in the 3D space, that's why in one fragment, it's possible that you still see this, okay, in one segment. Then what we did next is to do next generation sequencing. Remember, this is actually one piece of DNA, which means it's double-stranded. It has a positive and a negative strand. Then we can sequence the positive part and negative part. Then we can map it back to the reference genome. The point of doing that, or the purpose of doing that, is to find out where they are from. For example, I know this part comes from chromosome 14, this location, for example, here. And this part comes from also chromosome 14, but this location. This is the base pair location. For example, if you have chromosome 14 right here, as you can see here, okay, then that's the first base pair. This is the last base pair. The last base pair is 106 million. Okay, because chromosome 14 is very big, very long. Okay, then you have this kind of one of this kind of thing. That's called one contact. This is called one contact. If you keep doing this for the whole genome or the whole chromosome, you will be able to generate this kind of library. So this library contains all of the contact we know. For example, chromosome 5, this location, and chromosome 12, this location. This is called the inter-chromosome contact because it's really telling you the proximity between two different chromosomes, and very accurately tell you which part of the chromosome is in contact, which part of the other chromosome. Then if you plot this into this contact map, you will visualize something like this. It's a heat map. The more red color you see here, that means more contact you will observe from here. So you can generate this kind of a contact map, raw contact map. So what I want to tell you is that we did something similar on all the chromosomes. You can see the 2D contact map are different. So that means the 3D structure is different. Okay, and we plot the healthy people's chromosome 14 and also the 3 different types of cancer cells. You can see for chromosome 14, they are all different. So that means the 3D structure of this chromosome is different between healthy and the different types of cancer. And also this one shows you the contact map between different chromosomes or inter-chromosome contact. As you can see, the healthy B cell is different from the cancer cells. So that means the inter-chromosome contact patterns are also different. So what I have mentioned, everything is about population cells, which means you have a 10 to the power of 5 cells, and you generate a consensus data. Consensus like Hi-C experiment. But for this one, people can also generate similar stuff for single cell. So you just do similar things, but it's for each single cell. Okay. And then we got something for single cell to compare population cell or bunk cell Hi-C. This is for X chromosome. You have this many contacts, 923,000. For single cell, you only have 438 contacts for the same chromosome. So as you can see that for single cell, it's very sparse. You have a lot of zeros. So the first challenge for us, for computational scientists, is trying to, how do you build a 3D structure based on this? So this is a constraint problem. Basically, you want your 3D structure to fit all the constraints. It's easier to do on this kind of heat map, much harder to do on this one because it has a lot of zeros. Okay, so my lab built a model to do that. And we put the structure into a cube and do simulation. And we got a structure like this based on single cell Hi-C. As you can see here, every black dot is one Hi-C contact in a single cell. And then you can generate the 3D structure based on algorithm. This is for the X chromosome. That's another example showing the X chromosome, but we map a lot of epigenetic data, like the histomodification and this lamine enriched TAD, and also everything mapped into the 3D structure. And this one shows you the structure we generated based on extremely sparse data. As you can see here, there's only a couple of black dots. Okay, and we still can generate something like this. This is imputed data. That's why you can see a little bit better. This is high resolution, 50K base pair. This is for inactivated X chromosome. This is activated X chromosome. Okay, so then what we did next in my lab is to generate the genome-wide 3D structure based on single cell. This is one of the examples. As you can see, this one just exited the mitosis here. So if you remember, the cell divide, and it's just exited this mitosis state. So the cell is in this kind of V shape. As you can see here, this is a V shape. This is V shape. The 3D structure is generated by our algorithms. The Hi-C data is generated by a science paper. As you can see, in that paper, it tells the difference between paternal and maternal, and our algorithm works with that. And this one is another example showing you that you can see that it's just is a late G1 stage and mitotic. As you can see, the structure, 3D structure of the chromosome rapidly decondenses from the rod-like mitotic conformation like this, the rod, okay, because it's highly condensed to a more spherical structure as G1 progresses. Okay, so what next? There is something called a TAD, because as you can see here, this is the whole genome, and this is one chromosome. And people find out that in each of the chromosomes, it's formed up by each kind of small balls, and we call this TAD. This ball structure is conserved. Okay, I got 1 minute between human and mouse. So what are we going to do with this, with the regenerative and the non-regenerative? Because in the previous research, we generated the gene expression, chromosome activity, like attack data, histomodification data for both regenerative and non-regenerative. So my research will be looking at this non-regenerative and non-regenerative 2 types of neuron cells from the 3D structure perspective. We're going to see whether the 3D structure is different between the non-regenerative and generative neurons. And also we're going to plot this kind of graph. Each node represents one gene. If they are spatially close to each other, we can plot one edge. Then we can use graph autoencoders. The deep learning part comes from here to find out which kind of genetic and epigenetic features actually is most influential or the causing factors to generative and non-regenerative, so that we can make biological meaningful conclusion. For example, some genes expression or what kind of attack data actually cause the neuron cells to be able to regenerate. What kind of gene expression and his modifications actually cause the neuron cells to be non-regenerative? So that's the question we're going to answer using deep learning, specifically the graph autoencoders. This is my support. Thank you.
Thank you, Dr. Wang. Let's thank all the speakers again. Now we have about 10 minutes to do the panel discussion. I'm going to take questions from our audience. The first question is from Dr. Odelia Schwartz. I think this is for Dr. Joe's Principe. She said, I'm enjoying all the talks, Joe's. There should be a potential to utilize your information theoretic approach for studying or designing deep learning neural networks for understanding neuroscience. What are your thoughts about the links to the neuroscience?
I think it's a very important question. My view is the following. I think information theory has a great value. Information is something that you can act upon. You see, so if you are giving me lots of data, or if you give me a network or something, you never know how to act upon it. So information has this interesting concept, right, that we can act upon it. And so it is, and besides that, it's a scalar, right? So it's something that you can quantify. And so I really think that the most important aspect is try to pose problems in a way that you talk about independence, you talk about dependence, and things like that. Those are crucial elements, right, that I've seen in the presentations, and that and normally these aspects are built only in modules, but information can be used as cost function, right? And most of the times you don't need to create an error, right? You can compute dependencies between variables, data from two domains and things like that. So I really think that this can be very, very useful. Of course, remember that when we talk about mutual information on entropy, you have to be able to estimate the probability density function. This is the fundamental trick, right? The difficulty. But it's not a trick anymore. The science of creating estimators improved tremendously. Now you can even have networks that estimate mutual information for you. But I prefer, the methodologies that work with the data directly, because mutual information is associated with two data sets. And if I have the data, I don't need a network. I can estimate the entropy or mutual information directly. But
do you think in neuroscience experimentally, we usually have less data than modeling work? I mean, in modeling work, you can easily get distribution and compute dependency, entropy, etc. But usually in experiment, do you think that's a difficulty to apply?
It is. It is difficulty. But remember, neuroscience works a lot with time series, right? There's a lot of information in the time series, right? So the issue is, and you can estimate entropies and the mutual information on time series also, but you have to be careful. It cannot be just entropy. It has to be the entropy rate and the mutual information rate. And then all of a sudden you have all the time of your experiment. That is data that you can apply without using windows as everybody still uses. You get a window of data and then you compute things on Windows. Now you can do this sample by sample. So there are now very, very interesting techniques that can be brought very effectively to neuroscience. But so maybe we have to come up with a summer school or winter school. In Miami, it has to be a winter school. But we'll do a winter school on how to use information descriptors in neuroscience. And I think it will be a good idea.
That sounds great. I'm looking forward to that. Thank you. Our next question is for Yun, given by Linda. Do you plan to incorporate diffusion MRI data into your model?
We plan to do that. Actually, we are currently using the DTI data for using the free surfer to analyze the more, and it's kind of another side of work for the TM patients. But the DTI data will definitely be used in the analysis. And our final aim is to find out the kind of estimate the mechanisms of why this TM pain happens. So we will utilize all of our, not only for the pain recording data, but also for our resting data and also the MRI T1 images, also like kind of utilizing all of them, as we cannot get so much data for comparing with the common more imaging data sets.
That sounds great. So we run out of questions. Okay, we get a new one. This is from Stephen. So if they are so dynamic, how important is the specific structure for understanding cell FNX apart from mitosis or meiosis? Could structures be somewhat random? I think this is for Dr. Wang. Can you answer it?
Can you say the question again?
If they are so dynamic, how important is a specific structure for understanding cell FNX? I'm not sure what FNX is, apart from mitosis and meiosis.
If they are so dynamic, how important is a specific structure of understanding cell FNX apart from mitosis?
That means function. Function. Sorry, function. Right. Thank you, Fabian.
Somewhat random.
Could structures be somewhat random?
Could structure be some kind of random? Okay, it's a good question. I don't know. Yes, I think so. There must be some randomness there, but there must also have some non-randomness there, for sure. Because the hypothesis is that the genome 3D structure actually is helping the regulation process. Basically, you put the enhancer, which is far away, sequentially far away from the promoter, and then somehow it can fold and come to activate the promoter. That's one of the example. We also believe even between different chromosomes, probably there are some kind of roles different genes are playing. For example, you have a gene in chromosome 14, it probably got activated by another gene from another chromosome. So is there random stuff? I think so. Some of them, because it's like it's not like a metal structure. It's fixed. It's more like a tofu. It's shaking. So that's why there are some kind of randomness, but there are definitely some non-randomness, which is randomness in chromosome structure equals to noise entropy. Noise. Well, there are some noise. But for example, for the population cell high-C, the data actually is generated from 10 to the power of 5 cells usually on that level. So there is noise there for sure, because when people are doing a high-C experiment, it's not perfect. So there is some noise there, but we have computational method to remove the noise or bias. The easiest one is to use correlation. Actually, in one of the slides I show you. After you remove the noise, you can see the pattern much more clearer. So there is some.
Can I just add something here quickly?
Yeah,
I think there's a distinction between dynamic and random. Dynamic doesn't mean random. And while there's randomness in the movement of the atoms and Brownian motion in the movement of the macromolecules, the structures are definitely stable over time. So you have ensembles of structures that could exist in multiple states, but the average structure is stable, and it is usually, if the cells are synchronized and doing the same things, you will see those structures be the same in all those cells, and these are definitely control systems.
Thank you. I think we are toward the end of this session. I would like to thank all the speakers again. I think all 6 talks are very exciting, amazing talks, and the discussion also very, very exciting. So we are looking forward to seeing you next year. Hopefully we can do this symposium next year in person in Miami. I would like to hand my host to our next, my colleague, Rachel. I think she's here. I think you can take the control and move on your session right now. Thank you very much.